Namaskar. Today I'm going to discuss about cell cycle and the second part. First part, I have already discussed what is actually the cell cycle, what are the different stages of cell cycle, and I have clearly mentioned that what actually the phenomenon occurred in different phases of interphase, G1 phase, S phase, G2 phase, and M phase. And I have also mentioned in the previous video this chart, that is in which phase of the interphase and what are the different regulatory proteins present in different phases of the G1 cycle, S cycle, G1 phase, S phase, G2 phase, and M phase. Here I am again given the same chart for the understanding purpose, and we will look very closer today at the different cell cycle proteins, that regulatory proteins, how they are functioning, what are the nature of each and every one cell cycle protein. Now let's start that in a previous day the G1 is G2 phase, M phase, and in a G1 phase I have already discussed that there is a two kind of CDK, CDK4, CDK6, and along with the CDK, these two type of CDK, in a G1 cyclin D is present. Alone CDK is not able to perform the function of running of cell cycle from G1 to S phase. Alone CDK is not able to function. Cyclin is a partner. Here is a CDK and cyclin together. They work together and phosphorylates the different substrates. So while the cell run from G1 to S phase and cell pass the G1 S checkpoint. This is the major checkpoint of the cell cycle. The major checkpoint of the cell cycle is performed, or that is the major checkpoint is crossed by this CDK4, CDK6 and cyclin protein, and another CDK in a G1S checkpoint is present CDK2 and cyclin E. So these these three CDKs, CDK4, CDK6, CDK2, and two cyclins, CDKD and CDKE, they play a vital role during the initial stage of the cell cycle, that is the G1, when G1 is passing from G1 stage to the S stage. So we'll take the closer view on that, and some of the characters, the traits of the cyclins, what is actually the traits of cyclins and CDK. So step by step and we will discuss it. Let's see what actually the CDK and why it requires cyclin for their performance. Now CDK, as its full form is cyclin dependent kinase, that is, its name implies that it is a cyclin dependent. So for performing this CDK protein, it requires cyclin. So here at the first step I have mentioned here that a CDK alone is incomplete. Its active site, its CDK's active site is inhibited by its own T loop. Its own T loop inhibits its own active site. Now cyclin protein gets attached with the CDK and they separate the T loop from the active site. As after the attachment of the cyclin to CDK, CDK's conformational change occurs. CDK protein's conformational change occurs. So why? Its active site is partially removed, and in a second stage another friend of CDK is there, is CDK activating kinase. This CDK activating kinase phosphorylates the threonine amino acid, and as a result of phosphorylation of CDK's threonine amino acid, its conformational change takes place again and the active site is fully free. So why now, after attachment of CAK, CDK and cyclin is active now to further performance. So this is the CDK performance criteria of the CDK cyclin. So in the next stage here we will see that what is actually the inhibitor of cyclin CDK. Cyclin CDK partners have so many inhibitors. That is, when cyclin and CDK they perform together, cell cycle will run continuously. So it is as we need it that cyclin CDK works, but if they works continuously without any interruption, then the term tumor and cancer is attached to this. That is uncontrolled cell division. Cell will run and run and run and produce a uncontrolled and produce a wart-like structure or tumor. That is the main concern of our society and civilization now. So here in the next stage, the CDK cyclin and cyclin is performing in a wild stage. That is, they performing well. So cell will run and divide to the next cell. That is, the growth will occur. Now, what are actually the inhibitors of CDK cyclin? Suppose here is the active CDK cyclin, they will perform. But if cell cycle is required to stop, then another protein is there, that is UI1. It helps to phosphorylate the CDK. Two phosphorylation occurs at the CDK. So the active CDK cyclin pair is now inactive due to the UI1. This is the inhibitory protein, CDK cyclin inhibitors. And again, after inactivation, CDC25, this is a phosphatase. Phosphatase means they separate the inhibitory phosphate group from the active site. So why? The CDC is a phosphatase, and after the function of the CDC, the inhibitory phosphate separated and it again becomes active. That is, here after the entry of the CDC25, CDK cyclin becomes again active and start cell cycle. Now another inhibitor, these inhibitors are called CKI. It is also a CKI, CDK cyclin inhibitors. This CDK cyclin inhibitors is a P27. This P27 protein has so many functions. We will discuss later here. This P27 protein, when gets attached, it will destroy after attachment with the CDK cyclin. Its function is again inactivated. So as the CDK activating kinase is there, which that CDK activating kinase helps to activate the cyclin CDK complex. CDK inhibitors are also there, UI1 and P27. Here CDC25 is a phosphatase. It also helps to activate by dephosphorylation, that is by separation of two phosphate groups. Here it is again inactivated due to CDK inhibitors, which destroyed the active site. So this is the B point. And here another CDK inhibitor is there, the P21. We will later see the pathway. Actually, I am discussing each and every proteins and their functions. Now, P21 is another CDK inhibitor. These CDK inhibitors are so important, and this CDK inhibitor, this P21 CDK inhibitor is CDK2, CDK4 and CDK6 inhibitor. That is, this P21 inhibit in that area, that is CDK4, CDK6, CDK2. That is, these CDKs are very important for passing of this cell from G1 to the S phase. So this P21 is very important CDK, and these are tumor suppressor genes. Why I am discussing later. Now, the P21 inhibits CDK2, CDK4, CDK6. Also it inhibited PCNA. We know in a DNA synthesis, DNA replication, PCNA is a proliferating cell nuclear antigen. Proliferating cell nuclear antigen. It is a DNA polymerase association factor, associated proteins. It helps to function. It helps to function DNA polymerase and proper replication. Now, PCNA is also inhibited, so that this P21 inhibit DNA synthesis at the S phase. Also, not only this, it also inhibits the DNA repair system, but it also inhibits the DNA synthesis as well as the repair. But only exception is that nucleotide excision repair is not inhibited by the P21 protein. So P21 is direct inhibitor of CDK4, CDK6, CDK2 as well as PCNA. Another important protein is SCF. SCF protein inhibits the cyclin D and cyclin E, and it is actually a E3 ligase. Sorry, this function is 26S proteasomes. By 26S, by activating the 26S proteasomes, and like the APCC, that in a cell cycle, two this kind of 26S proteasome functioning protein is there, SCF and APCC, and SCF again inhibit P21, P27 and Y, all CDK inhibitors. So SCF is actually the friend of G1 CDK, that is the CDK cyclin pair. So these SCF inhibit the P21, P27, Y1, these CDK inhibitors. Another most important thing is that P53 protein. This is it is also known as guardian of the genome in vertebrates. That is, if single copy of P53, in future classes we will discuss details in P53 about the P53 and what is actually the character. This is the tumor suppressor gene, and this P53, single copy of P53, if damaged, it causes a syndrome, Li-fraumeni disease. Mostly in MCQs the questions are asked the Li-fraumeni disease. Single copy, if it is mutated, then it causes the disease Li-fraumeni disease, and at the adulthood there is a multiple tumor during the adulthood region. And in human beings, more than 50% cancer is due to the P53 mutation. What is the function? It arrests cell cycle in the G1S region. It starts apoptosis, and if DNA damage occurs, it stops cell cycle at G1S region. So here I have discussed about what is CDK, how CDK becomes activated by cyclin, what are the friends of the CDK cycle is the CDK activating kinase, what are the enemy or what are the which inhibit the CDK cycle, is V1, P27, P21. These are the inhibitor of the CDK cyclins, and also P21 is another inhibitor of CDK cyclins. Now how P21 function? P21 function by inhibiting the CDK4, CDK6, CDK2 inhibition, cyclin D, and PCNA inhibition, and also inhibit DNA synthesis phase. So these are the regulator of the cell cycle progression. If these proteins get mutated, that is the CDK inhibitors get mutated, then cell will run continuously, uninterruptedly, and so why these are oncogenes, and these all are oncogenes and proto-oncogenes. Proto-oncogenic functions they have. If they are mutated, tumor will start. So in the next class we will discuss in a details of these P53, P21, these functions. Thank you.